So, hello, this is Jonathan Agin, the Executive Director of the Max Cure Foundation. And as we announced earlier on some of our social media channels, we're going to be making some announcements in December, and we're excited about those. And some of the announcements that we're very excited about include some medical research grants that we're going to be presenting over the next week or so. And so today, I'm honored to have one of the recipients of one of the grants that we'll be giving out, and that's Dr. Oren Becher, who's joining me today. And MCF, Max Cure Foundation, has created a research mission that's focused on funding gaps and funding research projects that are aimed at clinical translation, meaning going into the clinic for children. We try not to duplicate funding and try and make our funding really impactful. A little bit about Dr. Becher, and then I'm gonna be quiet and ask him some questions and have him tell us a little bit about his work. But Dr. Becher is currently an associate professor of pediatrics, hematology, oncology, and stem cell transplantation and molecular genetics at the Feinberg School of Medicine at Northwestern University. He's a neuro-oncologist who's focused specifically on finding therapeutic pathways and new therapies for diffuse intrinsic pontine glioma, or DIPG. It's the same brain tumor that my daughter, Alexis, died from in January of 2011. In fact, I first met Dr. Becher in 2010 at Memorial Sloan Kettering when my daughter was on a clinical trial that he was running up there. So we go way back. And so I'm honored today to have him joining us and answer some questions. And so first, Dr. Becher, can you explain a little bit about the work in your lab and what you focus on, and a little bit more about your background?
Yeah, sure. Thank you. Again, well, first I'll start by thanking you for choosing to support our work. My laboratory focuses exclusively on DIPG, and we started working on DIPG way back in 2008, 2009, before I even had my own lab. When I was at Sloan Kettering, as you mentioned, I was taking care of some children with DIPG, and I realized that actually nobody was studying DIPG. So I decided that that this would be a good brain cancer to focus on, because nobody was really working on it, and there was no hope for a cure. And I felt that, you know, with good science, we may be able to change that. And so I trained with Eric Holland at Sloan Kettering for 6 years and learning how to make models, and then was fortunate to, with some of the lab members, to develop the first genetic model for DIPG. And this was around 2010, before we even knew about the histone mutations. And then I moved to Duke in 2010 to set up an independent laboratory focusing specifically on DIPG, and was fortunate to recruit some graduate students and postdocs that helped me build our various models. We've developed a lot of different subtypes of DIPG. Our expertise is that we can make a lot of different flavors of DIPG, as DIPG is not just one disease. There are different subtypes of it with very complicated biology. And so we develop different DIPG models using different cells of origin with different genetic alterations to try to understand what are the weak spots. What is a good target for therapy? Ultimately, the most important thing that we do is always thinking about how we're going to find some new drug that we can test in children with DIPG, and using the science to just help us prioritize which drugs, which therapy, which modality is most likely to work.
That answers the question, why DIPG? In terms of the tumor in and of itself, for people that don't really know much about specific childhood cancers or don't know much about DIPG, can you just explain a little bit about what is DIPG and what are some of the problems that you face trying to treat these kids?
Right, yeah. So DIPG stands for diffuse intrinsic pontine glioma. So it's a type of brain cancer that arises in children, and the tumor is located in in a part of the brain called the brainstem, and specifically the pons. And because I think the most challenging aspect about this tumor is because the pons also contains critical brain cells that regulate our breathing, our heart rate, our blood pressure. That means that a brain surgeon cannot take out this tumor because the tumor cells are sitting right next to these critical brain cells that help our brain work in these very basic functions. And so the tumor cells just grow in this very delicate location next to these critical brain cells. And so the challenge has been, how do you give any therapy that can only target the tumor cells and not the normal cells without, and I think that's been the challenge. And so actually the only intervention that has been shown to prolong survival by just a little bit for DIPG is radiation, focal radiation. And that was described in the 1960s. And so DIPG, again, it's just a little bit more background. So it usually arises in children around the age of 6 or 7, sometimes younger, sometimes older. And once it's diagnosed, the children only live about 1 year from diagnosis. And the standard of care, which I just mentioned, is focal radiation, was discovered 50 years ago. And even though we've been trying really hard with a lot of new clinical trials and new therapies, nothing has really worked to prolong the survival of these children. And radiation is the only thing that has been proven to have some efficacy, meaning it can slow it down, it can kill some of the tumor cells, but ultimately it's not a cure. And so I think the field as a whole now is trying to move the field forward. We've sort of been able to discover a lot of the genetic alterations of DIPG just because the technology has been available. But I think ultimately the challenge is, how do we use this new knowledge to find something that works for these children?
And that's kind of leads us into what we're going to talk about today and the grant that we're presenting to you. And so the reason that we're connecting right now is because the Max Cure Foundation, through the assistance of Connor Cures, which will soon become the Connor Fund, will fully become the Connor Fund at the Max Cure Foundation, is able to present research grants to novel and aggressive research efforts that are aimed at moving the needle. And so when I think about people that are doing research in DIPG or brain tumors, I've known you for a while, and I've known about your research for a while, obviously. You always come to the forefront of my mind in terms of people that are trying to use cutting edge science to move the field ahead. So with that being said, the Max Cure Foundation is, and again, through the benefits of the Connor Fund, through Connor Cures Foundation, is proud to present a check for $20,000 to support your research into DIPG. And if you could just explain a little bit what that money will be used for and what you expect or hope to expect will come from your research, I'd appreciate that.
Sure. Yeah. Well, thank you again for supporting our research. I think we're really excited about this project because it's utilizing one of the newest techniques that's received a lot of attention called CRISPR. And CRISPR is like really new technology that allows us to cut pieces of the genetic code wherever we want. And the reason we're excited about it is because we want to use this new technology to find the weak spots in DIPG. So the past decade, we've unraveled all the genetic alterations that give rise to DIPG, but we don't really know which ones are the most important to target in the clinic. I think, there's a lot of buzzwords like, can we target the histone mutation or some other genetic alterations, but we don't really know that. And so the idea is that if we can use CRISPR, which you basically delete all the different genes So we have about 20,000 genes in our cells, and a lot of them actually are not really well understood. Some of them are, but the majority are not. And I think the idea is, if you can, what we're going to do is an unbiased screen, use a library, and delete, each one of these genes and see which one of these genes is most important for cell growth. So we can assess, identify some new weak spots in the DIPG. And I think the reason we're excited about it is because it's an unbiased screen. So we don't know the results, and we have no bias. We're not focusing on the histone mutation or any other exciting gene. We're just doing a screen. We'll delete each gene in the DIPG cells, both in our mouse model and in human DIPG cells, and compare the results between the mouse model and the human cells and see which genes are most important for DIPG cell growth.
With the hopes of ultimately then finding therapies to match or to target. Target.
Yeah.
Semi-accurate?
Yeah, no, definitely. Once we identify the gene. Can you hear me?
Yeah.
Yeah. So once we identify the, now I have an echo. Okay. So once we identify the gene that's most important for DIPG cell growth, then we can look for the drug that does the same thing.
Got it. And if you could just briefly, I know as a researcher, one of the things that you spend quite a bit of time on, or you have a team in your lab, hopefully, that spends time on writing grants and looking for funding for different projects, funding from the NIH, funding from other sources. What role does a grant like the one we're giving from the Max Cure Foundation play in terms of supporting your research and supporting your lab?
No, I mean, it's amazing. I think, again, this is sort of like a high risk, high reward approach. If I went to the NIH and said, can you support me doing a CRISPR screen with no preliminary data, they would laugh, or they would not fund it. So, this is like a chance for us to explore a new technology. Like we don't have any grant specifically to do this unbiased approach with CRISPR in human and mouse cells. So I think this is like a new avenue for my laboratory that potentially can, move the field forward. I think in general, NIH and some of the more traditional grant agencies, they want to see a lot of preliminary data. They want to see that you've already done the work. So then they feel that they're taking little risk, with taxpayers' dollars. But I think, for curing a rare cancer like DIPG, where not that many people are working on it, we have to take more risk. We have to do these really new creative technologies and apply them to DIPG cells.
And how long do you think that it's going to take to do the work on this?
Well, we're hopeful to have meaningful results by next summer.
Okay.
And because then my goal is to actually take the results from this pilot study and submit it for NIH with preliminary data. So once we get some hits with the CRISPR, we found this new gene or a couple of genes that seem to be really important for DIPG cell growth, and we want to expand on it and maybe find some drugs to target that gene or set of genes, then we can apply for the NIH and see if they feel that we have enough data to get funding from the NIH. But sometimes it usually takes like a year or two before you generate enough data that the NIH would be willing to invest in it.
Got it. Well, we're excited to watch the results and to stay in touch. And again, we're honored to support your work. We're not there in person, so I can't hand you a check, and I don't know whether you'll be able to see this this big check here and how much it is.
Okay, yeah, no, I see it. Thank you. I appreciate it.
But that's our ceremonial check for you. We're happy to support your work and excited for these types of projects because, as you said, they're high risk and high reward, and we're honored to support what you're doing in your lab, and looking forward to hearing back from you and seeing what you find.
Yeah, no, thanks again. Like I said, we'll definitely update you. We're really excited to get this going. We're going to get started right away and update you as we progress with testing different DIPG models with this CRISPR screen and seeing what kind of results we find.
That's great. And frankly, by next year, summer of next year, if that's the target, we can talk in person when you run on the MaxCure Foundation Hood to Coast team.
That's right. Yeah, and that's possible. But yeah, well, like I said, I would definitely keep you up to date. But I am excited about the Hood to Coast, and I do believe that I will get to do it once at some point.
I'd love to have you run with us. That'd be great.
No, yeah, definitely. I'm excited about it, and I am just looking for the right time to do it.
Understand. Thanks, Dr. Becher. Appreciate it.
Thanks, Jonathan. Thank you again for the MaxCure, and thank you, Jonathan, for your support.
You're welcome.
Have a good day. Thanks.
I think we're off now, or well, we're still recording, but Erica's gonna
edit
magic and